question is called Medicine and Manure, Proving the Protein Functions of the pasture-phage ELE24. For this project, we worked with bacteriophages, which are viruses that solely attack and kill bacteria. And because of their ability to do that, they have proven to be useful in killing multi-drug resistant bacteria. Our phage was isolated from a dirt sample collected from a cow pasture, and then it was sent to the University of Pittsburgh for sequencing. And from that, after we got the sequencing back, we were able to identify the genes using various bioinformatic programs such as HHPRED and NCBI BLAST. And then those analyses revealed genes with unknown functions and genes with known functions.
So like she mentioned, we gathered our stool sample from a cow pasture, specifically my family's cow pasture. And then we put it in this conical tube and put some juice on top of it. That way the phages would come up and we could get them. We shook it really, really good. That way the phages would let loose of the dirt. Then we let it settle for a bit and then centrifuged it down to get all the juice off of it. We pulled up that juice, inoculated it with the host bacteria, and put it in our plate. And that was our direct isolation. And as you can see here, it produced beautiful samples. Two actually, which were both ours, ELE24, and Annapurna, which you can also see those posters. From there we took about 10 microliters, give or take, of phages from this one right here, and proceeded to do serial dilutions on it. And basically inoculated it with more bacteria, put it on more plates. We did that a couple times so we were sure it was just that one genome. Then we made like a web plate, which is basically just a bunch of phages on one agar plate. Put more juice on top of it, pulled that back up, figured out how concentrated it was. Then we sent it to our microscopist, Dr. Tajide, and that's how we got this cool picture. From there, we were kind of like, okay, we know these are separate genes, or at least we were reasonably certain. And then we sent it off to the University of Pittsburgh for the sequencing so we could determine the gene functions and such. And this is where Sallie steps in.
So now that they've kind of given an overview of what we did in the fall and kind of our goals here, I'm going to explain one of our genes as an example of this process. So this is one of the problem genes we had, gene 18, and this is one of the cases where a lot of the evidence pointed towards a whole in function, which is what we wanted to call, but we were not able to do that based on a small piece of evidence, so it was a really hard call to make. So first of all, we put the segments of the gene into these bioinformatics programs, and so that's a lot of the figures you see here. So HHPRED is the first one we used, and this essentially tells us the crystal structure of certain functions and how that aligns with our gene. So HHPRED showed a 98.28% probability of a whole in function, so that's what this red is right here. That is a really good probability, and so it shows it's very likely that this gene has a whole in function. And the same thing with NCBI BLAST, that showed the whole in function in other homologous genes. So they both had really good probability with that function. And so because of those high probabilities, we're thinking, okay, let's look and see what we need to have in order to call that function. So in order to call a whole in function, according to the protein call list, which is like a spreadsheet we used to figure out what we needed, there were our two transmembrane domains, which is required for this. So that's what these two things are. And then synteny between homologous genes, that's shown up here. So we compared ELE with Timothy, and as you can see, it aligns really well. And we also had another indolysine in the area, which is this purple block here. and that's another requirement we need for Holen. But where we run into our issue is in order to call this a Holen, there cannot be an additional transmembrane domain in the area. However, there is one, and that's what this green area is. So because of that additional transmembrane domain, we cannot call this a Holen. We must call it a hypothetical protein or a transmembrane protein. So this is a good example of, even though a lot of this evidence, a lot of the strong evidence points towards the Holen function, you cannot call it that because of the protein call list.
And so just a good brief overview of everything that we do. After we've been able to call a start and a stop for all of our genes, we take the DNA segments and put them into our bioinformatics program, such as HHPRED and NCBI BLAST, as we've talked about. Then we're able to interpret this data to assign a call. So like we use HHPRED to see it most likely looks like a Holen, but we have to compare it to our other systems to say, actually, we can't call it that. And then we look at our homologous genes. So a lot of the times we use Timothy just because they are so similar, and we're able to kind of base our calls off of what they've called. And then we have to look at our protein call list. So this is like a master sheet that has all of the proteins that we could possibly call, the requirements for why and why we can't call them. So yeah, that's kind of just our process of how we do this. In the future, we'd like to perform host range studies to see if UE is interacting with other types of bacteria. And we'd like to investigate the hypothetical and unknown proteins, such as what we've called here, gene 18. We want to know what actually is going on, what actually this protein is doing, because it's not just doing nothing. So those are kind of our future steps for what we'd like to do with our gene.